Okay, so thus far we have been talking about B cell development. Last time we went through, we sort of reviewed some of the details about B cell development, where we saw our pro B cells, our pre B cells, and the things that they're doing. And then we also saw the immature B cells. Remember that the immature B cells are characterized by the fact that they actually have a BCR on their surface. So you can see it says here IgM expression, or MIgM expression, membrane IgM expression. Fancy way of saying that they have a B cell receptor on their surface. And we also talked about kind of the things that an immature B cell does. And so what were the big actions that are happening with an immature B cell? Yep, Sebastian. Yep, so we're going to test for self-reactivity. Krishna.
It's also testing the light chain?
Yep, so we're going to test the light chain, and we're going to test for self-reactivity. And we usually think about those in this order. And if a B cell that has developed is self-reactive, it has 3 choices. There are 3 things that could happen to it so that we can avoid having autoimmune disease, so that we can avoid self-reactivity. In general, these are the 3 central tolerance mechanisms. So what do we got, Jamie?
Deletion, receptor editing, and anergy.
Yes. So we've got these 3 options. We can either have deletion of the cell, so basically that cell dies. We can have receptor editing, which is where the cell is going to make a new light chain so that it changes the thing that it is specific for. And we also have anergy. And we left off talking about anergy last time. So what did I tell you? What's anergy? I should probably say a little bit more about anergy before I get into the rest of this. So here's my anergy slide from last time. Here's another anergy slide. See, look, deletion, receptor, editing, anergy. So what's anergy? Yeah, Sydney.
You get like a low-level signal through the cell because not as many B cells react to self-antigen, and the B cell gets deactivated, but it's still allowed to keep the bone marrow.
Yes. So in both of these cases, we're getting a pretty high signal from that, say, membrane-bound antigen. The B cell gets a lot of signal. And that B cell is either going to die or start being reactive to something else. If, however, we have a somewhat lower signal, that B cell is going to be energized. It's going to undergo anergy. And what does it mean that a B cell undergoes anergy? What is an sometimes we call these B cells anergic, or they've been energized. What is an anergic B cell? Just make sure we're all just real clear on this, all on the same page here. Yeah, Sebastian. Kind of, yeah. So we usually talk about anergic B cells as actually having something happen so that their signaling doesn't work anymore. You can't turn them on. These are sort of permanently off cells. So these cells are unresponsive. As I mentioned, they are present, but they are not functional. So these cells are allowed to leave the bone marrow, but they're turned off to not do anything when they leave the bone marrow. So there is a frequent question that comes up here. What question might you have when seeing this information about these anergic B cells? Yeah, Sydney. Why? Can we kind of expand the why? Yeah. Why do we keep them? What's the point of keeping a cell that you're not going to use? Why do we keep this cell around? So if we look at our 3 options here, we can pretend there were 3 cells. This is only showing us 2 cells. We can pretend there is a cell that did deletion, and a cell that did receptor editing, so pretend there's 2 cells there, and a cell that went for anergy. Of those 3, are any of them like more or less dangerous than each other? Like which ones would be like any that would be like really bad versus less bad to have go into the body? Yeah, Jamie.
Do you mean comparing like the high and low, or the low?
Yes. But before we get here, up at the top, which one's the most dangerous? The ones that are binding highly. They bind to some self thing strongly. In these experiments, they're binding strongly because it's a membrane antigen. But in fact, the B cell doesn't know if it's a membrane antigen or not. It just knows it got a lot of signal. It just knows it binds something strong in the bone marrow. If it binds something strong in the bone marrow, it's really going to be a bad guy if it goes into the rest of the body, right? It has some potential for disaster. What about this one? Is this one as dangerous? So it's not as dangerous, but it's still kind of dangerous, right? We still don't really want it around, but it's not as crazy dangerous as the other ones. What would be the downside of setting up a situation where you were really strict here? You deleted anything, you got rid of any B cell that even like looked at you funny. What would be the downside of that? Yeah, Rishi. Okay, yes, you're weakening your immune system. How are you weakening your immune system? Krishna.
Wouldn't you be turning off more B cells than needed?
Yep. So what would that mean, Jamie?
Because each B cell makes a specific antibody, and only that B cell makes it.
Mm-hmm.
If you're turning off any B cell with any cell reactivity, you're going to be turning off the creation of a lot of antibodies.
Yeah. So let's imagine, this is not exactly how biochemistry works, but we'll pretend it's how biochemistry works. So we can say that all of our B cells are somewhere on this line between being self-reactive and not self-reactive, right? Some of them are really, 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 really self-reactive. Those guys are bad. Some are not self-reactive at all. Those guys are good because they protect you from the SARS-CoV-2s and the Ebolas and all the other pathogens, right? So the idea here is, well, where do we draw the line? How your immune system's kind of making a line here. Let's imagine your immune system was really strict and it drew the line right here. Anything that was more even a teeniest bit more self-reactive, like towards that way, we killed. And we only kept those guys. Let's imagine that that was the case. What might be a problem here? And it's a really hard line. You kill every single one of those cells. Yeah, Rishi. Yeah, you are going to be in a situation where you're not going to react to very many things. You only have a really small number of things that you react to. Maybe this one right here was the one that you needed to respond to SARS-CoV-2. But it had like one amino acid in common with something in self, so you killed it. Well, then you're not killing that virus. Sad for you, you're dead. Right? So if you're too strict on this point, if you kill cells that shouldn't be killed, if you are like, oh, don't want to get, that one might be a little too self-reactive, then you could end up with too narrow of a repertoire of B cells. Not enough B cells to actually protect you, right? So now what happens if we have a super hard line, but we decide that our super hard line is going to be right there? We kill every B cell that is this much self-reactive, and if you're over here, you are fine. You don't get killed. Jamie? Yeah, you're probably going to end up with some autoimmunity. You're probably going to end up with some of these B cells that are going to start to attack self-tissue, and you're going to have some autoimmune disease. So you can see this is kind of a problem, right? The way that this actually happens is that we kind of have this sort of, we kind of set it up like this, where there's different areas. If that binding is super strong, if we get a lot of signal, that's really dangerous. And this is where we get rid of the cell, either by deletion or receptor editing. But then there are these middle cells. And these are the cells that end up being anergized. They make it out of the bone marrow, but they're off. Except there are situations where they can be turned back on. When I think about the situation where they are turned back on, I think about like the immune system sledgehammer. Like something crazy has to be going on. There has to be so much inflammation and so many signals. Like your immune system has to be like going nuts. Okay? So under basically normal circumstances, they don't turn on. But if everything goes crazy, they can get turned back on. They have to get basically like a sledgehammer's worth of signal, not just a little normal amount of signal. And that can actually be really useful. So let's imagine you have some cell. Let's imagine that you get infected with Ebola. And you don't have a good B cell to combat Ebola. Because that Ebola B cell was here. You're going to try to combat Ebola. You're going to have like your normal old responses to Ebola, right? And Ebola's going to win. And you're going to get really sick. And then when you're really sick, your body's going to try to call a Hail Mary. And sort of, you're going to have so much inflammation, you're going to have so much going on, you are going to feel bad. But that's going to actually start turning on some of these cells. Sort of like, okay, absolute last resort. Will any of these help me? Absolute last resort. Maybe these cells that were sort of medium, that were not quite the super dangerous ones, maybe we got to try to use one of those. Because these weren't good enough. So we see anergy in both B cells. We'll see anergy in T cells later on, where the cells are, you can sort of think of for all intents and purposes, off, unless like craziness happens. And if craziness happens, then you call them in as kind of your last absolute resort. Yep, Sydney.
Does exposure to certain toxins cause that response, like with chemotherapy and stuff?
It could. It usually needs to be a pretty sustained inflammatory set of inflammation in order to turn those cells back on, but in theory it could. And so it's a little bit hard to sort of really get at the answers to that, because this is one place where things seem to vary a little bit from person to person. Like how sleepy are my cells versus how sleepy are your cells? And how easy is it to wake up my cells versus your cells? And so the idea is that it's really, like I said, it's a ton to turn that back on. And so you should not think of that happening under any normal circumstances, only like crazy circumstances. But how crazy is crazy might vary from person to person's immune system. So does that make sense? Some of you guys actually may have heard of, and we can talk about this more, some details like something called long COVID, where some people who are infected with SARS-CoV-2 have long-term effects, or some people seem to have some long-term autoimmune types of things going on. How could this be related to that? There is no data. We are clearly in the realm of hypothesis right now. But how could it be related? Yeah, Jamie.
Once those cells, those emergent cells get turned on, I don't know if they get turned back off.
They might get somewhat turned back on, or somewhat turned back off.
But if they don't get fully turned back off, then you're going to have some level of self-reactivation going on.
Okay. So that turn on, that sledgehammer, that's done by cytokines. So you might turn on all the ones there. Some of those might be helpful for COVID. Some of them might be autoreactive. You just turn them all on by throwing cytokines at them. And so you might wake up an autoreactive cell when you're also waking up that COVID cell. If you have gotten to the point where you had that sustained inflammation, where your immune system kind of went for broke, where your immune system was like, it's better than you dying. And so we could imagine that sort of the excess response might be turning on sort of this autoreactive response. Although, again, there's not total evidence on that. People are trying to figure that out right now. But I'm just hypothesizing how these things could be related. Yeah, Jamie? Potentially, yes. So chronic fatigue syndrome is a syndrome that has been out there medically for a while. And I will be totally transparent. There are a lot of physicians that don't believe it exists and think that the patients are making it up, or it's all in their head. And there's a lot of negativity towards those patients. But there's just been this idea for a long time. Suddenly, we started to have this long COVID disease that looks an awful lot like chronic fatigue syndrome in a lot of ways. And most of the chronic fatigue patients are like, yeah, I had a viral infection, and then I never really got totally better. That's what started my chronic fatigue. And suddenly everyone's like, wait a minute, what if lots of viruses could do this, not just one? And the chronic fatigue patients are like, hi, we've been talking to you for 20 years. You wouldn't pay attention to us now, and now you think we're cool. And that is actually kind of what's going on in the field. And so, yes, potentially this could be something similar going on with other viral infections. But again, this is sort of this is what people are actually doing experiments on right now, is to figure out what in the world is up with this. But it could be the same thing with chronic fatigue. Okay, so that's our Anergy. So now we can kind of talk a little bit more about, that successful B cell, and what's going on with that successful B cell. If that B cell gets to the point where it is making a successful heavy end light chain, and it's not autoreactive, now it finally is going to be able to leave the bone marrow. And when that cell leaves the bone marrow, it now gets to be called a mature B cell. Siri knows a lot about this. So we now get to have this mature B cell that leaves the bone marrow. One thing to note is this mature B cell turns off. Our mature B cell has no RAG expression, and that RAG expression is off for the rest of the B cell's life. We are never going to have RAG again. in this B cell. So there is one other kind of thing that we can notice about this cell when it becomes a mature B cell. If you look, the immature B cell is shown as having a B cell receptor on its surface that contains IgM. When that B cell is in the process of leaving, sort of by the time it is out of the bone marrow, the mature B cell is making both IgM and IgD. And so those are the first two. You can actually see that on this figure from your textbook here. Our B cell sort of coming out of the bone marrow is just making IgM, which is the one that's shown in black. By the time it's a mature B cell, it's totally out. It is making both IgM in black and IgD in red. If you recall the constant regions downstream of our VDJ region, the constant regions encoding IgM and IgD, constant region areas, so these exons, are the first ones. So the M and the D, the mu and the delta, are first. So M and D are first, and they are the first ones made by the cell. And in fact, the only thing that we really understand about IgD is it's a good way for us to be able to tell the difference between immature and mature B cells. Because I told you before, up until recently, IgD had absolutely no known function, and we only ever used it for distinguishing stages of B cells in development and finding what it was a mature B cell. And so you can see again in this figure from your textbook that the immature B cell right before it leaves just has IgM on its surface. The mature B cell, when it gets out of into the periphery, has IgM and IgD on its surface. So we need to talk a little bit about how that cell is getting both IgM and IgD on the surface. This is related to something known as class switching. And class switching is how we can put the different constant region sections paired with our VDJ heavy chains. And so our VDJ regions. So you can see that we've got the same variable regions in red that are pairing with these different constant regions, either mu, delta, gamma, or epsilon. And each of these are encoded by different regions, which are also shown here. Okay? Now, if you look, there is something important to notice about the genomic organization that is shown here. So at this point, we have maybe done VDJ recombination. And so V, D, and J are right next to each other on the DNA. We've cut out the intervening DNA. But we've still got DNA between the J's and all of these constant region segments. And you can see the amounts of DNA that we have between each of these areas here. So what do you notice as you look at these different amounts of DNA, these different distances of the DNA? Rishi? So you can see they're getting a little bit smaller out here, sure. There are a couple other things to notice. Yeah, Jamie?
There's a really distinct change in size when it comes to M and then D, and then for the next one, the change is much larger.
Yeah, so M is pretty close to VD and J, 6.5. And D is pretty close to M, 4.5. And then there's a giant space, 55. And then we have big spaces still. They're not as big as that 55, but they're certainly bigger than those two. So the two shortest spaces are right there. That's important because when RNA polymerase comes along and does its job, it's going to start transcribing at V. It's going to transcribe V. It's going to transcribe D. It's going to transcribe J. It's going to transcribe mu. It's going to transcribe delta. And then it's going to get tired. It can't go 55 whole more kb. Like, all this together wasn't even 55 kb. It can't go that far. That's too far. And so we can actually make an RNA that has VDJ, MD, but we can't make an RNA that has any more of this stuff because it's too tiring to go that far. And so, in fact, what happens is this is the piece of RNA that gets transcribed here. We have our VDJ region, we have the mu region, and we have the delta region. And so at the beginning, those B cells are transcribing this whole region and making this whole RNA. But they don't make G, E, and A because they're too far away down here. The RNA polymerase is tired. It can't go that far. Then when this RNA is made, the cell can splice that RNA two different ways. One way to make a version with M, and one way to make a version with D. So this cell has the DNA that's letting it do M and D, and it's doing this by alternative splicing. So the DNA doesn't get messed with right now. We're just looking at changes in the RNA. Similarly, if we look at the fact that our B cell is both making a B cell receptor and is making an antibody, this is also coming from that same mRNA. There is a place where we can get the transcript to end right after M and make a little hydrophobic segment. See, the pink goes with the pink. Or we can have it end after M and make this transmembrane region. The green goes with the green. And we can do the same thing for D as well. And then we can splice differently that RNA. And so, in fact, we can get 4 different RNAs off of this one DNA. We can get secreted IgM. We can get membrane-bound IgM. We can get secreted IgD, and we can get membrane IgD, all off of this one piece of RNA. And so this is sort of a summary of what we have seen happening to the genes encoding the B cell receptor or antibody section so far in the B cell's life. So we've seen the V, D, and J's come together, which also helps us move the transcriptional stuff. Both of those things are places where we had to deal with the DNA. In fact, honestly, numbers 1 and 3 are 2 different ways of phrasing the same thing. So both of them are just, we broke the DNA, and we threw some out, and we pasted it back together. Let's imagine we do not like that. Can we go back to the beginning? What do you think, Karen? No. Once you cut the DNA and throw some out, you can't get it back. Once you make that change, it's irreversible. You're done. That's you and all your progeny got that new DNA. And so those are 2 irreversible changes to the cell's genome. When we're doing that adding and subtracting, or that cutting and pasting, we can add and subtract base pairs with junctional diversity. Again, this is happening to the DNA. We're adding and subtracting new base pairs to the DNA. This is also irreversible because we're changing the DNA. However, alternatively, the cell is also going to be able to make IgM and IgD, and it's making that change based on how it splices RNA. The DNA is not getting touched in this process. And so this is something that the cell can kind of swap back and forth. And this process is regulated and can be sort of changed because we haven't messed with the DNA. Similarly, if we want to switch between making secreted antibody and B cell receptor, this is happening at the level of RNA, and so we can reverse it, and we can sort of regulate this process. But now we can also think about the rest of that B cell's life, and some of the things that are going to happen to that B cell. And so now we're moving into the whole body that's not the bone marrow, the place where we can see microbes, the place where we can see antigens. Immunologists call this the periphery, or sometimes the everywhere else that's not the primary lymphoid organ is basically the periphery. So now our B cell is out, and it's going to be looking for antigen, which is what you can see happening on the right side of this slide, where our B cell is looking at peripheral tissues. I am going to give you an overview of some things that happen in the periphery, though we are going to talk about them more later in the semester. However, just for full transparency, when I give you this overview, I am technically telling you about a particular type of B cell. In reality, there are a bunch of types of B cells, and I'm really focusing on kind of the most famous one. So we're going to see lots of types of T cells later in the semester. I am largely going to talk about B cells, as if there's not a million types of B cells, but there's also a million types of B cells. My little sister did her PhD on B cells, and so if she were here, she would yell at me for not giving B cells their due and all of the different kinds of B cells. And so technically, we are actually going to be talking about cells that are part of the B2 lineage that are going on to become mature B cells. But in fact, we're kind of talking about this process right here, where we see the cell that it goes up here. But there are, as you can see, there are other paths that B cell could take. So there's one called a marginal zone B cell. There's one called a B1 cell. We're really officially talking about classical B2 B cells, or classical B2, the classical B2 B cells. Your textbook also does mention this information. So there are marginal zone B cells. There are B cells. They vary a little bit in terms of some proteins on their surface, some details about exactly kind of how they're working. And so really right now when I'm saying this is how the B cells work, I am officially talking about follicular B2 B cells. So just for full disclosure, that's what we're doing. There are other kinds of B cells. Now my sister will not yell at me. And if we kind of have an overview of what's happening to these B cells once they've left the bone marrow, they are going to go into some kind of, typically they're going to be hanging out in secondary lymphoid organs like the spleen or the Peyer's patches or lymph nodes, where they're going to be searching for microbes, potentially finding them and attacking them. And so this is kind of the details of what's going on here. You can also see this in this figure from your textbook, where these cells, number 1, 2, and 3, are going to sort of around looking for their antigen. They might find that antigen. And when they find that antigen, they will divide to make many more copies of themselves, as well as change. And so their functions will differ a little bit. You can sort of see that here. You can see the cells kind of getting some slightly different shapes, things like that. So there are a few different kinds of changes that will happen. There are also some changes that happen to the antibody that is made by those B cells. And so there are two major changes that happen to the antibody made by a particular B cell when that B cell is in the periphery. Those two changes are called class switch. That's a bad piece of chalk. This is a slightly better piece of chalk. And affinity maturation. And so these are two changes that can happen to this B cell when the B cell is in the periphery. So I'm going to focus first on class switch. And so class switch is how we can change which type of constant region we're using. We could take this IgM and we could switch to using IgG. And so we are going to think about how does class switch happen? Because these classes are sometimes called isotypes, sometimes we'll refer to this as isotype switch as well. So how does class switch or isotype switch happen? And so this is one of those things that, this is one thing that, like I said, sometimes drives people who've thought a lot about biochem stuff nuts when they first hear that it happens, because they're like, no, that's not how proteins work. But I promise, I'll tell you how. In fact, I'll tell you how right now. You've actually already seen an example of class switch. And that was how that B cell early in its life could make both IgM and IgD. That's actually it making both more than one isotype. And the idea of what was happening there, if you recall from like 2 slides ago, is that we have one big RNA that has the information for the M constant region and the information for the D constant region. And that RNA can alternatively splice. So the same VDJ can be paired with either the M constant region information or the D constant region information. And so we can make a heavy chain with this variable region plus M or this variable region plus D, just by alternatively splicing this different RNA. So this is sort of class switch, although this is not the thing that people think about as frequently as this sort of other piece to class switch. Because oftentimes we like to think about the other isotypes. IgD doesn't really have much of a function. So I just showed you how we could make IgM or IgD. Hooray. IgM should be easy because it's the first one. So we don't really even have to do anything to make IgM. Hooray. We can make IgD by splicing, like I just showed you. Hooray. But who cares, because IgD doesn't do anything. But then we can also think about how we get IgG, IgA, or IgE. And those have some interesting functions. Those are some we might want to make. And we have this problem in making them. So what's our problem in making them? Again, from like 3 slides ago. Yeah, Sebastian. Yeah, those constant region DNA pieces are really far. We can't make RNA polymerase go all that long way. That's too far. You can't just make a transcript and splice it. RNA polymerase is not able to make that long of an RNA. It's just not. It's too tired. I can imagine it of me trying to run a long distance. Can't do it, too tired. And so the way that we are able to make IgG, IgA, or IgE is that this cell actually has to do something called class switch recombination. And so class switch is done by class switch recombination. We've already heard the word recombination once this semester. When do we hear the word recombination this semester? Yeah, Jamie. We heard about VDJ recombination. Recombination is basically where you break and you cut and paste DNA. You break the DNA and make it paste it back together again. So in fact, for class switch, we're going to have to cut and paste the DNA again. In front of each of these little constant region areas, there's something called a switch site. We could actually have a recombination event at a switch site and delete the intervening DNA. So if we want to make IgG, for example, we do a recombination between those two sites. We cut out M and D, and then G suddenly is right next to the VDJ region. And so now we can make VDJ G, because we cut out and threw away M and D. And so you can sort of see that idea here. And so now we're going to get that G region close enough that we can actually do transcription. If a cell switches to making IgG through class switch recombination, can it go back and make IgM? Nope, because it just threw that DNA away. So this is another situation where we are making a change to the DNA and where it is not reversible. So this cell is going to actually completely change its DNA again. One other thing that you might realize is that this probably doesn't involve the enzymes RAG, because I told you that RAG is off for the rest of the B cell's life. So this has to have some other recombination method. And we will talk about that other recombination method later in the semester. We can also think a little bit about how affinity maturation happens. And with affinity maturation, our B cell is going to actually change how its antigen binding region is set up so that it can actually get stronger antigen binding. So you can see we go from kind of okay antigen binding to really good antigen binding. So the affinity, the binding strength, got better. It matured. That's what we call this affinity maturation. So this is also a change that's going to happen in the periphery. Just like with class switch, this is something that only happens after we see antigen. And so we might wonder how affinity maturation happens. Affinity maturation happens because of a process known as somatic hypermutation. So somatic is a word that means body cells, basically. What do you think hypermutation means? Yeah, Rishi. Mutating rapidly. What were you going to say, Justin? A lot of mutations. And so, in fact, what happens here is that we get a lot of mutations in these cells. And if we actually look at those mutations and we graph how frequently there are mutations, all of those mutations are in the V and J part of the light chain and in the VDJ part of the heavy chain. So basically, we get a whole bunch of mutations directed right there. And that is, and in fact, if we even look in more detail, we zoom in even more to look at the heavy chain and the light chain, we can see that those mutations mostly all come in those complementarity determining regions, the CDRs, which were those little loops at the end of the immunoglobulin domain that come off the end of the heavy and light chain and touch the antigen. Those, all of the mutations are in the base pairs that encode those loops. So the base pairs that encode the loops are shown in like the pink, and where the mutations are is shown in the red. And so you can see that in this sort of example antibody response, we picked up a whole lot of mutations, and they were in those antibody combining regions. And so that is going to let our antibody change its structure. We change the DNA, so that changes the amino acids, it changes our protein, and we're going to get stronger binding for antigen. However, or just in general, somatic hypermutation and class switch both happen pretty late in a response. They are both things that are generally seen in a secondary response. So later in a response, we tend to see more IgGs. We tend to see higher affinity because affinity maturation has happened. And so we really are only going to see these things kind of late in our response. Going to skip ahead for just a second. And I could imagine that you might say, okay, I want to know more about the details of how these things work and exactly what's happening with somatic hypermutation and class switch. For a B cell to do hypermutation and class switch, there are a couple things that need to happen. One of those things is that the B cell actually needs to see antigen in the periphery. So these are things that happen after the B cell has seen antigen in the periphery. However, there's another thing that has to happen for this B cell to do these processes. And that is that the B cell has to interact with a T cell. I can't tell you any more about the life of our B cell until I tell you about T cells. Because our B cell relies upon T cells for many of the rest of the parts of its life. And so we're basically going to leave our B cell here. We're going to be moving into T cells. This is why I sort of set up where the exam is and everything. So that we can then see the whole T cell story, and then we'll come back and put our B cell and our T cell together. And we'll be able to see our B cell doing these processes. So just as a summary, this is a table from your textbook talking about all of the things that we have seen that are important for B cell activation and how we are generating B cell diversity. So we can use multiple VD and J genes as well as light chains. We've got our heavy chains. We depend on RAG1 and RAG2. We add P and N nucleotides. We can't put together multiple Ds because that would break the 12-23 rule. We use allelic exclusion so that each B cell only makes one receptor. We can secrete an antibody as well as keeping the receptor on the surface of the cell. The different constant regions give us different functions. And we can have this somatic hypermutation after we stimulate. So this sort of summarizes everything we've talked about thus far. This is, of course, a table from your textbook. What you might guess is that we are covering up another half of it about T cells, but we haven't learned that yet, so that's why we don't have to look at the part yet. And some of the answers will be different. And we can also kind of generally summarize all of the potential changes that can happen to the immunoglobulin gene. So we have this irreversible change, because it's happening at the DNA level, that is involved in our VDJ recombination, which also changes the locations of promoters and enhancers. We have this irreversible change to the DNA that gives us P and N nucleotides, that gives us additional diversity. We could actually have M and D made when that cell is first leaving the bone marrow based on RNA splicing, and so we can sort of change that as we need to. We can also change whether or not we're making a secreted protein or a membrane-bound protein by changing the RNA. So again, that's reversible because neither of these things are affecting the DNA. They're both happening at the level of the RNA. And then we're going to have somatic hypermutation and class switch, where again, we are going to change the DNA either by point mutations or by a recombination process. And so again, these are going to be irreversible. These are happening in the periphery later on. This is all happening in the bone marrow in the primary lymphoid organ. Okay, so that's my B cell stuff.